Okay, and we are back. So welcome to everybody. This will be the first episode of our fourth season of the International Legume Society webinar. I welcome everybody. I'm Nessuno Vicari from Italy, and from today we have an announcement, and we start with an announcement, and then we start with our speaker. But I just need to remind you that our next conference is coming up. It will be held in June in Dubrovnik. You find all the information on our website, on the dedicated website, and the call for paper, call for abstract is already out. So if you want, you can already submit your abstract. I will keep reminding you about the conference in the next coming months. But without further ado, I welcome our speaker for today, which will be Dr. Fanjiang Kong. He's professor at Guangzhou University, and he's an expert about soybean and all things, all factor about flowering modulation. So today we are going to have this very interesting webinar on the genetic and molecular underlying mechanism about soybean photoperiod. So Fanjiang, please welcome, and
yes,
the mic is yours.
Okay.
Sure.
First of all, I would like to thank this community to inviting me to give a talk, which I be able to share our recent works to the community in the world. So in our lab, our research area focusing on the photoperiod flowering, and lately we're also doing a little bit about plant architecture, which related to the yield, which today I will cover these two topics. So my topic is around, it will be about genetic mechanism of photoperiod flowering and the plant architecture. So my name is Fanjiang Kong. I'm from Guangzhou University in south of China. As we know, photoperiod had been found 100 years ago by two scientists from the United States, Professor Garner and Allard. And together, they used soybean as a model plant and find all the plants have the photoperiod, which can be categorized to two types of photoperiod response: long day plant and short day plant. And soybean is a very, very typical short plant and very sensitive to the photoperiod. And after 100 years, but we don't know what the genes control the photoperiod response in soybean and what mechanisms for the flowering and the response. So you can see we have one cultivar from the middle of China, which in the middle of China, that's a medium long day, so about 14 hours of the long day in the natural light. So this will be flowering about 40 days. But if we move this cultivar to the high latitude, more high latitude in north of China, and this is the long day around 16 hours of a day, and they will flowering very late to 60 days, to 20 days later than compared in the middle of China. In the other hand, if we move this to the north, to the south, to the short day, they are flowering 20 days earlier compared with the middle of China. That means the only one genetics, but if we move different photoperiod areas, they will show very different flowering behavior. So we are very interested for this phenomenon, and this also very important for the breeding, because if we want to use the cultivar from the southern area to the northern, you will have no flowering, and you will have a long time to select the progeny from these hybrids. So if we know the mechanisms, we can easily to manipulate the genes to control the right time for flowering and to have the higher yield. And we have been doing this identification of the genes and the study of the mechanisms of these gene interactions. We have several publications. So due to the time limitations, today I would like talk a few works related for the flowering time. So soybean is the one of the crops had been originated or domesticated from China, from mid of China, around 35 to 40 north latitude degree. So and then 500 years ago around, and then soybean had been spread to the world for several thousand years, and to the north to Korea, to Japan, and to the Russia, and to the south to the India or to the South Asia, Indonesia, and then later and the soybean has spread to Europe and to North America and South America, and recently also has been cultivated in Africa. So this dissemination require very more genes to control this wide adaptation. So the first of our interest is came from the whole soybean adaptation to the tropics, like in Brazil. So this is the map of Brazil. So Brazil is a tropical region, very hot and very short day, around 12 hours of light and 12 hours of night of the day. So at the beginning, so the cultivation of soybean in Brazil, they had introduced the late flowering cultivars from South America, from Indiana state and Louisiana state, but they are still early flowering, around 20 days or 30 days. And then during this cultivation, they make a lot of process using late flowering plant, and they able to find two genes and control late flowering in the third day. So one locus called the G locus, another one called the E6 locus. These two genes, if they have a mutation, they will show very late flowering and late maturity. Brazil can have a normal plant, a higher yield plant. So these two genes very important for the soybean cultivated in the shady area for South Asia, for Southeast Asia, and for South America, Brazil and Peru, or the place. So we are very interested for these two genes. So we collect several natural mutations, carry these two mutant alleles, and we make crosses and we are doing the mapping. We first map the J locus to the chromosome 4, and we're doing the positional cloning and fine mapping, and we map to 4 chromosome around 200 kilobase in the number 4 chromosome. We able to identify the genes as the candidate genes for the J locus is early flowering 3 genes in Arabidopsis. This gene is a very famous genes for the circadian clock. So we have 2 mutants. Both mutants have mutations in the EF3. They have a loss function mutations. And then lately, we also map the E6 locus also to the chromosome 4 region, the same region as the J locus. And then we clone this E6 gene. This E6 also a gene of early flowering 3, but different alleles. So the E6, they have the mutation is about in the early flowering 6 genes. They have have insertion of the usual transposon in the exon 4, the same early flowering 3, but different area at J. So we confirm both J and E6 are ELF3 genes. So it means ELF3 gene is very, very potent for the plant in the short day adaptation. And then we collect many short day ladder races and accessions, and we sequencing about the ELF3 genes. We found many mutations, nature mutations, at least 8 or to 10 mutations. All these mutations is located in the tropical regions from India, from Myanmar, from Thailand, from Peru, and from Brazil. And all these genes have the loss function mutations. And we confirm the J mutation or E6 mutation is very important for soybean to adapt the short day areas, the tropical areas for the South America and South Asia. And this nature mutation had been occurred many 2,000 years ago in the South Asia and 100 years ago in Brazil. They have 2 independent adaptation centers or mutation centers. One is around here, one in South Asia, one is in the South America, mainly in Brazil. And literally, we figure out this EF3, the J is the EF3. They are components of evening complex in arbutus. So the evening complex had been identified and confirmed by Professor Steve K from the from California State University. And these 3 proteins, they are binding together in the evening to control the plant circadian clock in the evening, and they make a loop. So we are wondering how this complex working for the flowering time or flowering sensitivity in soybean. So we try to knock out this complex, and we found this complex requires the transcription factor of LUX, LUX, to binding the downstream genes. So in soybean, there are two LUX homologs. We call LUX1 and LUX2. So we have a double mutant. So if we have double mutant, all the complex had loss functions because all the complex like EL4 and EL3 and LUX, they require this transfer factor of LUX to binding downstream genes. If this have the double mutant show the fully loss function of evening complex, and we phenotype this mutant. We have a single mutant, we have double mutant, and to see the behavior of flowering time in the short days. In the short day, the single mutant, either of LUX1 or LUX2, they have no phenotype. But the double mutant show very late flowering, about 60 days later than the water type, 2 months later. So they flowering about so the water type and short day they flowering about 20 days, but the double mutant show 80 days, 6 days later than the water type. And we continuously to cultivate the double mutant. They show the very strange behavior, growth behavior. They can, and we cultivate until 280 days. They still survive. They have the flowering in the top, and they make new branches, a new shoot in the top. They have the pods in the bottom. So this plant is similarly, this plant changes from the annual plant to the biannual plant. They continually make the new shoots and new flowering and the new leaves to continue the growth up to 4 meters high, very high. It seems this the evening complex is very critical for this plant. When the evening complex have the mutations, the plant growth behavior changed differently. And then we also make the plant growth and the long days of the evening complex mutant, and they also show the same flowering time at the short day. So this you can see the column, the last column. They also show the same flowering behavior. That means the evening complex is a very critical sensor of a photoperiod. If the evening complex disappear, they will they cannot sense the flowering. They cannot sense the photoperiod, and the consequence, they flowering at the same time about 80 days. So that means the evening complex is a very critical components or genetic factors for the soybean flowering time, also for the photoperiod sensitivity in soybean. And then we did a lot of biochemical protein interactions to see the mechanism why the single mutant have no flowering time, but the double mutant show very severe late flowering phenotype. We found 3 proteins, LUX1, LUX2, and J. They can interact each other to form homodimers or heterodimers. And finally, we're able to make a model for the mechanisms. We very brief, very easily, very simple model. So in the A column, so there are hetero complex of LUX1, LUX2, and the G. And they have the early flowering. But in the column B, so if either of LUX1 have a mutation or LUX2 has a mutation, they will form the homo complex. So you cannot see flowering time difference. They will same flowering at wild type. But if the J have mutations, so this complex will lose the suppressive activity. They will show late flowering. But if we have the transcriptor binding cannot have binding the gene downstreams, so to completely lost the function of the evening complex, they will show severe and very late flowering. So this is a very easy mechanisms to explain the evening complex have a critical functions for the flowering time, or also for the for photoperiod sensitivity. And then we make a new name for this evening complex mutant. We call this the second picture. We call the Guangzhou mammals to reminding the photoperiod discovery. They use the Maryland mammals. This is the tobacco, also used as a model plant for the identification of the photoperiod. So the Guangzhou mammals is the evening complex mutant. They also show very high plant stature and more leaves and continued growth, and show the photoperiod no sensitivity in soybean. And the photoperiod sensitivity is very important for the plant for the cultivar in the high latitude regions, like in Russia, in northern Japan, in Korea and northern China. Because if the cultivar have the very strong sensitivity, they will show very late flowering, will have no seed. So in the northern areas, they require the plant show the how to say insensitivity to the long photoperiod. So and then we want to identify the cultivar which show the photoperiod insensitivity. And we collect a lot of local cultivars, and we use a facility artificial long day. And if the two set of the cultivar they show same flowering time, we treat this cultivar as photoperiod insensitive cultivars. Use this method, we are able to identify 29 cultivars that show the photoperiod long day insensitivity. And then we use the genes, use three genes control the photoperiod sensitivity, E1, E3, and E4. E1 is a legume specific transcription factors, and E3 and E4 is the fat from A2 and A3 in soybean. Either of these three genes, either of two genes have mutations, loss functions, the soybean can cultivate in the high latitude. If these three genes, either of two have mutations, this cultivar can adapt to the high latitudes, show photoperiod insensitivity. But these three genes combination has been already used for the cultivars in America, in Canada, and in south of China for the cultivars to cultivate it over there. We already knew these genetic mechanisms, but we don't know the molecular mechanisms. We know the E3, E4 is the fat from, and the E1 is the transcriptor, is downstream of the fat from, but how the signal is trans-targeted from fat from to the transcription factor. We thought they require the circadian clock in between to transaction of the signal from fat from to the transcription factor of E1. And then we made several mutants of we make a double mutant of E1. We make a triple mutant of a double mutant of E3 of E4 and the triple mutant of E1, because in soybean there are three homologs of E1, E1 like A and E1 like B. We make a triple mutant of E1 and double mutant of E3 and E4. We show the transcription of E1. We found the transcription of 3E1 strongly require the two fat chromes, E3 and E4. If E3, E4 have mutations, these three transcripts no longer expression, and they also in the same pathway. And then we check the fat from A, E3 or E4. They can interact with the LUX transcript in the evening complex of LUX transcript factor, and they can make a degradation of LUX to release the suppression activity of the evening complex. And in the meanwhile, the phytochrome A protein can also interact with the E1 directly to stabilize of E1 protein, to make sure the E1 not be able to degradation by other factors. And then we make a model of this pathway. So from the beginning of the photoperiod receptor of phytochrome, they interact with LUX to degrade the degradation of evening complex, release the E1 expression. And at the same time, the protein can also to double check to interact with the E1 protein to make sure the protein can be stabilized to suppress E1 active expression. So this work firstly to connect from the beginning of the phytochrome, circadian clock, transfractor E1, and flourishing of FT to make a new pathway in soybean. And lately, we also want to know how the evening complex protein can be controlled by other factors. We found the E2 E2 are also a GI of the arabidopsis circadian clock genes. E2 protein have 3 homologs in soybean: E2, E2 like A, and E2 like B. We first make a triple mutant of E2, and we cross with the wild type of Williams 82 to make a single mutant, double mutant, and a triple mutant. We found, so this E2 also very important for the photoperiod-insensitivity. You can see the figure B, the last column. So if the E2 triple mutant, they show very early flowering, both on the short day and the long day, the same flowering time, but no also no photoperiod sensitivity, means the E2 also very important. So we wonder how does the E2 protein how the interaction with the evening complex, because evening complex previously we found evening complex very important for the photoperiod flowering, for the photoperiod sensitivity. And then we found the E2 can interact with the evening complex with the J, with the EF3 protein, and the degradation of EF3 protein. And E2 can also interact with the FCAF to bring FCAF to the J and to degrade of EF3. And then lately we make a cross. We want to know, because LUX double mutant show very late flowering time, no sensitivity, but E2 triple mutant early flowering time also show no sensitivity. But what we want to know, if we cross these two mutant to make this five genes mutation, how they flowering behave. And we found, so this 5 mutations, the flowering behavior in between of these two kinds of mutations, so they not like E2 triple mutant, and not like LUX mutant, but in between, means they can make a feedback regulations. So we check the expression. We found the J or LUX, they can inhibit E2 expression, and this protein can binding to the E2 promoters to down-regulate E2 expression. So finally, we can make a model. This is the feedback loop control the photoperiod sensitivity in soybean. Like in the second figure in the SD, so we make two complex, the morning complex E2 and the FCAF, and evening complex EF3 and the LUX. They are binding together. In the short day, the E2 have no functions, so the evening complex have a strong function to inhibit the E1, make early flowering. When we move this plant to the long day, to the most left figure, show the E2 in the long day. The E2 have function. They can interact with FCAF to make EF3 degradation, to release the evening complex, and to have more expression of E1, show the late flowering. So this the feedback loop perfectly explain the soybean, why soybean in the short day have a early flowering, but the long day they have a late flowering. If you have interest, you can read this paper we published last year in Development Cell in details. And then I will skip a few slides. This slide showing arabidopsis. In the arabidopsis, also the evening complex show the photoperiod insensitivity, but they use the epigenetics to binding with several 3-sense chromatin remodeling factors to control flowering time and photoperiod insensitivity in arabidopsis. And finally, we can make this kind of pathway for soybean from photoperiod A1, A2, and A3, and interact with the EF3 and the LUX, and together with the FCAF and GI to make a big complex to control expression of E1. And then E1 can control FT and to make this flowering pathway in soybean. This photoperiod flowering pathway or photoperiod response is very important for the soybean development, because from the germination to the shoot and to the leaf development to the plant architecture, all the controlled by the photoperiod. And lately, so from the rice and the corn also found the evening complex important for the photoperiod sensitivity in rice and corn by different laboratories, and means the evening complex, the EC, is very important for crops, for rice, for maize, and also for soybean. And because the E2 is very important, so when we're doing the GWAS or genetics, so all the flowering time populations can be found the E2 locus. E2 locus is a major locus, so many smaller locus cannot find. So we divide E2 to two subpopulations, small E2 population and the large E2 population. So to remove the effect of E2, we can identify the new flowering locus of top 9. Top 9, we clone top 9 is new genes of DTL2. So DTL2 is also blue light photoreceptor, and is also E3 ligase can degradation the interaction proteins. So E2 can interact with the DTL2 or top 9 and to promote E2 degradation to change the expression of E1 to make the flowering time. Also, FCAF also can interact with the E2, but the FCAF is stabilize E2 to make a opposite function of CAF and the DTL. So these two FCAF and DTL2, they are same families, but they have different or opposite functions to control E2 stability. We also found the selections of E2 DTR mutant and FCAF mutant in the long day cultivars. In the high we can see in the high latitude cultivar they select mutation of FCAF, but in the low latitude cultivar they select the mutant of DTL2. So these two genes control the photoperiod flowering, also control the yield in the either in the high latitude or in the low latitude. And finally, I would like to talk about how the plant architecture or the plant heat, plant internode control for the dense planting. So the green evolution used in rice and wheat, they use the very short plant. The genes, green revolution genes like SD1 or IHT1 in wheat and SD1 in rice. So, but in soybean, there are no this kind of genes. So we want to identify the genes. We generate a big mutant library, use the gamma ray radiation. We found one mutant called RIN1, reduce internode 1. And this show reduce the 40% of the plant heat, only 6% of the compared with the body type. We make a mapping population where we clone this gene, RIN1 gene. RIN1 gene is the SPAR protein, SPAR3 protein from soybean. SPAR is the protein is very famous for the in adaptation for the photomorphogenesis to control the light dependent plant growth, control the gibberellin pathway. So we are we generate the SPA3A mutant, and they show the short plant. And we identify the real one protein, SPA3A protein can interact with ATF1-2 transcription factors to promote ATF1 or 2 degradation, and then AT1 and 2 can binding to the gibberellin synthesis genes in the gibberellin synthesis pathway to control the gibberellin by synthesis to control the internode length growth. So, and then we use the mutant. We use different population sites to see the yield performance. You can see we use 250,000 plants per hectare to 450,000 plants per hectare. And you can see in the high more in the column, you can see the right in the mutant of ring 1 can increase the yield about 40%. The one gene can increase the compared with the wild type cultivar. And then we make a new cultivar. We call this cultivar use the ring 1 alleles. We make a cultivar we call DS89 cultivar in the northern China, and we continually to make a demo field about more than 3 hectares in 3 years. So this very high yield compared with the wild type also can increase the yield about 40 or 50%. So we in China we use more for the acreage. So 280 kilogram per mu calculated to the hectares around 4,000 kilograms, 4 tons, and to 4.5 tons per hectare. It's a higher yielding in soybean. And then this work had been highlighted by very famous Professor Chu. He's doing the green revolution in rice, and he mentioned maybe this ring 1 gene is the green revolution genes in soybean. You can use these genes to introduce to the soybean and to make a soybean for dense planting for more than 400,000 plants per hectare, even more, even 600,000 plants per hectare to increase the population size and to can increase the yield very much. And then finally, I would like to conclude our works in 2 aspects. First one, we find the Golden mammals, we find the immune complex and the E2, the GI genes, GI protein, and the immune complex play very critical roles in soybean to control the flowering time. We define the flowering pathway in soybean as from A to GI interact with the immune complex, and then this complex control E1 expression and E1 control FT expression and to make flowering. And this also the mutant of GI or EC can show no photoperiod response. This GI and the immune complex control soybean photoperiod sensitivity and photoperiod response. And the last work we found maybe a green revolution gene of ring 1 to use these alleles to introduce to the allele cultivar can increase soybean population size for the dense planting and to increase the yield very much. And then finally, I would like to thank our group and many collaborators from China and from different university and different research organizations. And also we are in preparation for many grants from AIFC, from CAS, and from the MOA. And last we sent this our group, so we have 40 students in our lab, and we are doing the soybean genetics and also soybean developmental biology. And thank you. I'm happy to answer your questions.
Thank you, Fanjiang. It was a very interesting presentation full with results. There's any questions? I see that already I have a question from the chat. I will read it for you. From Tom, yes. Are the RIN1 plants shorter than white type? This is why you need high plant intensity. So was it clear, the question?
You mean the E1 plant is very short, is right?
Is it shorter than the white type? So the starting white type?
Yes, the E1 triple mutant is too short, so we cannot use the E1 mutant directly for the breeding. So we need more high nodes, because the E1 have a few nodes, so we need a very more nodes. So we need the RIN1 have the, because also E1 mutant is very early flying, only 20 days. So in the normal cultivar, we require 40 or 50 days for flying, because we need enough vegetative growth. So the E1 cannot, because E1 only 20 days. So only 20 days they will change the phase from vegetative to reproductive. So this give us very less yield. It's not a good one for breeding.
Right. Maybe I guess Tom was referring to the RIN1, RIN, to the reduced internode mutant.
Yeah, the RIN1 reduce the.
Just to be sure.
Yeah.
Okay.
Yeah, reduce the internode length, also reduce the node number.
Sure. Okay.
Yeah.
There are a couple more questions in the chat. Just let me check them. Okay, so question number 1: will the recording of so this is about with the recording of this webinar? Yes, it will be in probably tomorrow on our YouTube channel, and it's also on our website. So as all the previous webinars, it's public and recorded. Then a question for you, Fanjiang, about E2 locus. How large are the effects of E2 homologs, so E21A, E21B, on flowering in northern latitudes? So very technical question.
Yeah, this the homologs of 3E2, the E2 have very big effect. The two homologs have very small effect. Even the double mutant of E2A or E2B, I think they only have 5 or 7 days earlier compared to one type. So this homolog have a very small effect, very small.
Okay, very small.
I answered the first question for the professor marketing. So the E1, because the RIN1 very short, only the plant in the field only about 70 to 80 centimeter, very short.
Okay.
So and then we can make a very high density planting, because if the plant is high, if we make this planting, they will have a large effect. They will also drop the pod or flower from the downstream nodes. But if this short plant, we can make a dense planting. They will maintain. They don't launch, but also they maintain the pod and the flower underneath the nodes. So this gene is very good for dense planting for the higher yield breeding. Yes.
Great. There are any other questions from the participants or in the chat? Doesn't seem so. So Fanjiang, thanks again. It was very interesting presentation packed with results, and it was a pleasure to have you as the opening speaker for this our new season of the ILS webinars. The presentation will be online in a few hours or tomorrow at last. And thank you to everybody. And just okay, no, just people thanking you on the chat. So that's it for today. I think I stopped the recording and thank all the participants.
Okay, thank you too. Thank you to give me this great opportunity to share our works, and let's keep contact.
Sure.
And
sure.
Maybe see you in the conference. Okay.
Sure. See you everybody in June. Hopefully.
Okay.
Bye.